My name is Kayla Crouch, and today I'll be presenting on direct RNA sequencing with Nanopore technologies. Okay, so direct RNA sequencing with Nanopore technologies is a sequencing approach that allows RNA molecules to pass through a nanopore sensor. In this technique, the RNA is ran through a pore and an ion current is produced based on what nucleotide is being read. Then it is converted into an RNA nucleotide sequence, and then each read includes exons, UTRs, nucleotide modifications, and end modifications. The methods for this approach includes extracting your RNA and isolating it, performing quality checks by checking its length, quantity, and purity, fragmenting the RNA, size selection, and performing adapter ligation. Then you run the sequence, and once you get the sequence, you can begin mapping and assembly. Controls that you can use for this approach include positive, negative, replicate, and RNA modification controls. A positive control would be running a known sequence to see if the sequence matched the intended results. A negative control would be running a sequence with no RNA to ensure false reads aren't getting picked up. A replicate control would be repeating the same sequence to ensure the results are reproducible. And then the purpose of running an RNA modification control would be to assess the sensitivity or accuracy of RNA modification detection. So this technique ties into our next generation sequencing module, and this can be directly compared to the Oxford Nanopore sequencing using DNA or cDNA that we discussed in class, and also the Illumina RNA-seq that we discussed in class. Advantages of this approach includes it eliminates the need for cDNA. So in this approach, cDNA is not necessary. And here reverse transcriptase, is optional. You can work directly with RNA without making it into cDNA. And here you don't have to work with transcripts that are difficult to perform the reverse transcription. And then next, PCR bias is eliminated. So PCR is not used in this method, thus you're not experiencing PCR bias that is commonly seen when using the Illumina RNA-seq. This method can provide information on RNA modifications, which are important in understanding the function of RNA molecules. And long RNA reads can be sequenced. I found this image or figure from a paper I'll be discussing in the next slide. This image shows a comparison between direct RNA sequencing and Illumina RNA-seq. Here you can see in the intergenic regions in the first graph, the Nanopore had a mean read depth at a twofold difference and was still higher in the other two to the right of that. And the authors concluded that this is likely due to a poly-A selection and intrinsic biases in reverse transcription and amplification steps of the Illumina RNA-seq protocol. And then disadvantages of this approach, these can all be compared to the Oxford Nanopore technology sequencing we discussed in class that uses DNA or cDNA. So here, this approach has a lack of multiplexing. It lacks 5' N-terminal recognition. It has lower throughput, and it is on average more expensive. So this is a study that the last figure came from, but this paper is direct RNA sequencing on nanopore arrays redefines the transcriptional complexities of a viral pathogen. And in this paper, the author used direct RNA sequencing to profile the herpes simplex virus type 1 transcriptome during reproduction infection of primary cells. And this figure shows a good example of the methodology and approach in a real study. And in the first step, a poly-T adapter is ligated to poly-A tails and used to prime the first strand synthesis of cDNA to stabilize the RNA strand through the nanopore, and this is seen in the second step. This is important to point out that this is optional and that some people do this in their methods, the use of cDNA to stabilize the RNA. And then in the third and fourth steps, the poly-T adapter allowed the ligation of the motor protein that is required to guide the RNA strand through the nanopore, demonstrated in the bottom step. This study looked at the 16S and 23S rRNA strands of E. coli that have 36 different modification sites and 17 different structures. They were looking to determine how metabolic and cold shock stress impact these 36 rRNA modifications using direct RNA sequencing. And using this approach, they produced results for all 36 of the sites seen in the blown up figure on the right. In doing this, they also proved that direct sequencing for more than 16 RNA modifications in one strand is possible. And if you look at the figure on the right, this shows changes in the E. coli rRNA modifications that were identified after metabolic and cold shock stress. These changes were color-coded in white, black, yellow, red, and green based off of t-tests of the RNA modification level changes between the normal growth and stress conditions. Lastly is an article that looked at the architecture of SARS-CoV-2 transcriptome using direct RNA sequencing with nanopore technologies. And this figure shows just that. If you look at part A, the leader represents the viral reads that have the 5' end sequence. The no reader displays the reads that do not contain this. Nuclear displays reads that match mRNA from the nuclear chromosome. Mitochondrial reads represent reads from the mitochondrial genome. And finally, the control represents the control RNA that was used in this procedure. And then the unmapped 4.7% shows the percentage that wasn't able to be mapped. And as a result, from this, the authors found that the complete SARS-CoV-2 genome was almost covered using this sequencing method. 12 nucleotides were missing from the 5' end, which represents the 4.7 unmapped percent. And the authors concluded that this was due to the direct RNA sequencing incapability to produce the 5' terminal end, like I mentioned in the disadvantages slide. And attached are references that I used to pull the figures and also just get information on RNA sequencing. I hope you guys enjoyed. Thank you.